So back to what we're talking about is the genetic diversity of rose rosette virus isolates, which is a project that I got involved in with John Hammond and Ramon Jordan, who you've heard both of them while we were setting up, and Venura Harath, who has been a long-time collaborator of mine in some bioinformatics studies. We began this project with the question of helping the breeders in understanding what are the isolates and what are the differences amongst isolates of rose rosette virus, if any. So while they are breeding for resistance, is there a chance that there's a resistance breaking strain out there? There's no information, and most people have been operating under the assumption that there isn't any diversity and there's just one strain or one kind of rose rosette virus. So I kept hearing this question a lot and decided to try to dive in with John and Ramon and Venura. And so this was our starting point, and this is where the RRD website with the diversity of sequence of genomes or diversity of across the US was a helpful starting point in getting conversations with Kevin and with John and Ramon about how rose rosette is reported nationally, and is there maybe a difference between rose rosette in one part of the country from another part of the country? For most viruses, there are geographical differences that result in mutations and diversification of species within species. And so the wonderful part of Ramon and John being on the East Coast and me being in Texas meant we were able to combine our resources in our different locations and come up with a wonderful study. So at the time we began talking, the SWDE and the RFDC, DE for Delaware, DC for Washington, DC, were two new genome sequences that Ramon and John were sequencing and have the complete full genomes of and are now reported in NCBI and in the publication that came from this study. And so we were able to compare that with sequences. And what we did different from other folks was we concatenated the full genomes. Rose rosette virus has 7 genome segments. Instead of comparing them individually, we could put segment 1 next to 2, etc. Concatenated them into one entire genome and compared them to identify different lineages. And this tree in panel C here is the outcome of that comparison. And you can see that there are different lineage groups within this tree. And so these are all marked here from 0 through 7 through 8. And the arrows, the blue arrows, point to the Delaware and DC isolates that Ramon sequenced. And you can see they're in different trees or different branches of the tree. They're not closely related as we would have thought given their close locations. And so with that, we went on and did further analysis of the genome differences. This is a statistical approach looking across the concatenated sequences. We looked at 95 genomes that were published in full in the NCBI database. There are more than that reported, but they're not complete genomes. There's maybe sequences missing from the 5' end of a segment or the 3' end of the segment. So we pared down our information to everything that was an entire genome and something we could validate as a unique individual. So we have 95 sequences, and the S is the total number of polymorphic sites, which means changes. So out of 95 sequences, there were 1,648 changes across those sequences. And then the total number of mutations was higher, 1,700 mutations. So you have 1,600 changes, 1,700 mutations, which means there may be one site in a genome, one nucleotide that has multiple changes. Okay, so that's why the mutation rate may be higher. Okay, so we broke that down by RNA 1 through 7 and looked at the polymorphisms and the numbers of mutations. And you can see in each case, the mutations are higher than the polymorphisms. So that suggests that there's a higher mutation rate than expected. And these are all the statistical values that are reported from the Tajimi, the Fuli, D, and F tests that shows that there are significant differences between genomes. And the greatest differences here at the nucleotide level is in RNA 5. Here we go. There's 209 changes, 223. And then RNA 7 here. These have the highest statistical rate shown. And so the neutrality tests is the significance of negative indicates that the mutation rate is not constant. And so this suggests a population expansion. Okay, so we, again, another way of viewing this data was looking for the potential for genome reassortment of segments. Looking at the concatenated sequences, we compared everything and looked for where maybe you have two different isolates infecting one plant, and RNA1 from isolate A and RNA1 from isolate B switched in the progeny. So that would be recombination. So possibly with 7 segments from isolate A and 7 segments from isolate B, you can create a new strain by the mixing of the segments. This is well studied in influenza virus. And so here identifies the breakpoints of those sequences. These black peaks here identify 3 major breakpoints across the this virus, and you can see this one, for example, is at the junction of RNA1 and RNA2. And so another breakpoint is at the close to the junction of RNA2 and RNA3. Other junctions are between RNA4, RNA5. So it looks like there's a potential for different strains coming together, or different isolates coming together. I don't want to call them strains yet. Different isolates coming together and mixing and coming out with maybe some new variations from changing segments between isolates. Okay, so to study that a little bit more, to present this in another way, we put together a rooted tree, maximum likelihood tree of just RNase 1, RNase 2, RNase 3 here. 4, 5, and 6, 7 are on the next slides, but let's look at 1, 2, and 3 here. If you just look at the architecture of the tree and the branches, you could see that, that there are differences in how the isolates group. Now, I put a yellow box to identify isolates. So obviously, you're not going to read every individual 95 isolate there, but the yellow boxes point to the DC and the Delaware isolates and their neighbors. And so RNA1 in the middle of the tree is the DC and the Delaware isolates. And then at the bottom of the tree here in group 2 is where they occur. And then in RNA3, they separate. They're in very unrelated groups here and at the bottom there. So if we look at RNA4, 5, and 6, similar happens. Here they're at the top of the tree. RNA5, they separate again into 2 different parts of the tree, and RNA6, they separate again into 2 different parts of the tree. So it looks like there is evidence that segments may be recombining, and that if you have a mutation in one segment, it could end up resulting in drift and movement into new isolates and new variations with exchanges amongst isolates. So that was interesting to know. This is a mechanism that may actually be happening. Then we looked at RNA6 quite closely here, and RNA6 was very interesting to all of us that were working on this project. So RNA6 encodes 2 proteins, 6A and 6B. So across here, say, is the RNA sequence, and the open boxes shows you the proteins. And they're overlapping proteins, and the nucleotide position, start and finish of each is presented. And this line here highlights a region between 220 position and 520 position. which we blew up here at the bottom of the slide. And it was an interesting region because it has a lot of U's in the RNA. So A, C, G, U. There's homopolymeric tracks of U's, extensive. And when we looked at that region, there were a lot of interesting changes. These 14 viruses were identified as unique isolates that did not recombine with any other. So when we did our recombination analysis, we identified all the individuals that looked like they could have been recombinant progenies. We discarded those and came up with these 14 as non-recombinants. And in our analysis, there were some clear changes in RNA 6, where there's a transition from a G to a U or a U to a G. There's some very specific nucleotide changes that were quite remarkable in their pattern. And for ease of the reader, we identified them as regions 1, 2, 3, and 4, with some specific CDU changes. So what does that mean? These mutations are individual mutations. As we broke them out here in our different regions, this is to highlight, for example, up top here, there's a lot of writing to do this. Where there's 7 U's in this region 1, in this isolate from Michigan, there's 8 U's. This is just one nucleotide difference, but that can change the whole reading frame. That could be a shift if it's overlapping the gene. Over here, you could see that there's gaps indicated by dashes and changes in the numbers of U's, where here there might be 6 or 4 compared to 5. And so amongst these isolates, we found difference by 1 nucleotide in different positions. So what was more interesting as well is where it's identified in red is where, say there's a mutation in region 1, the same isolate had a compensatory mutation in another region. So you might create a frame shift, but then it's corrected in another region. So this was intriguing to us that there was some real mechanism of changes happening. So we went from not knowing if there was any differences to saying that there's some mechanisms of a +1, -1 shift happening. When we look at the amino acid, these small changes in RNA 6 actually resulted in differences in the 6A protein. When we so here are 25 examples from the 95 sequences that are the best examples of differences we could find in RNA 6A or open reading frame 6A. Some of them are small proteins of about 31 amino acids. Some of them are longer amino acids of around 82, 85, even up to 92 amino acids, just because of those single changes in the homopolymorphic region of RNA 6. Quite remarkable. Most of the sequences end at amino acid 62 in an isoleucine. So the majority of RNA 6 open reading frame A's look like this one here. So there are differences. We don't know what this means to the function of these proteins or to the diversification or what that could mean in symptom expression. These are all wonderful new questions that we can begin to ask based on this research. Does a longer 6A protein have a different symptom outcome or a different disease outcome or a different host range outcome? These are kind of our jumping off points now that we have this information. So looking at the patterns of amino acid changes is important to identify different virus isolates that can be distinguished serologically as well. So if we use antibodies for ELISA tests, these informations can help us understand when antibodies may detect or not detect the presence of a virus. So if you have PCR positive results and ELISA negative results, perhaps we can really use this information to improve on our antisera development for diagnostics. These tell us about sites that are informative and changes that are not informative. So if we look at the RNA1, which encodes the RNA-dependent RNA polymerase, there's 31 changes that are informative changes, which is about 1% of the genome. If we look down here at RNA6B, there's 7 changes, which goes up to 3%. RNA5 has up to 6% changes. RNA7, or protein 7, has up to 5% changes. So these are very real and I think quite useful information. The nucleocapsid has the least number of amino acid changes. So that's a quite stable protein. So if you were going to make an antibody, I would pick one to the nucleocapsid protein to be sure it picks up the most different isolates. Less than 1% changes. But it also suggests that there's different evolutionary pressures acting on these different proteins within the host or within the vector. And again, this opens up new interesting questions. To really highlight what these changes look like, we overlapped our understanding of the amino acid changes into these maximum likelihood trees. And instead of showing the tree in a linear, I decided to show it circular so you could see it a lot better. The initial trees that I showed you a couple slides ago were the nucleotide levels. These are amino acid levels, looking at the amino acid changes encoded by the RNA-dependent RNA polymerase. We used color coding for the informative amino acids and featured them across the tree. And it's really interesting how at the beginning or at the bottom of the tree, there's aggregating patterns of amino acid changes. And then as you move further up the tree, some of these changes don't exist. They look more like the consensus sequence of what you all know. And so we could, across sequences, start to look for here, RNA2, there's again an aggregate of quite a large number of changes in a few isolates at the bottom of this tree. And then as we move further out, and we tried mapping these according to geographic locations. Some of it was easy, but again, most of our isolates that we had reported in NCBI, a large number of them were reported to be from Arkansas, although we couldn't confirm that they were from Arkansas specifically. That's the information that we're going on, and many of them were dispersed. So we looked more carefully at what was happening with our DC and our Delaware isolates, since our team reported those sequences as well. And here, the nucleocapsid protein, what's interesting is there's only 3 different versions. There's 1 amino acid change or 2 amino acid changes of a valine to an isoleucine, or a valine 61, valine 93. It could be valine 61, isoleucine 93, or isoleucine at position 61 and valine at position 93. Really no changes happening at that protein. And so some of these proteins have extensive changes. Some of these proteins are highly stable and not subject to evolutionary pressures. So we can begin to examine some of these questions. This shows you RNA, where am I? The remaining RNAs here. We've got protein 6B, protein 5A, protein 6A, and protein 7, and each of them have very different patterns of changes. With the 5 protein has the most remarkable aggregate of mutations of anything that we've seen so far. So if I were to suggest something is a symptom determinant or a potential for a resistance breaking mutation, I would focus on open reading frame 5 and RNA5 for its contribution to stability of resistance going forward. And then here again is RNA7, remarkable, or I should say protein 7, remarkable high number of amino acid changes, not unlike protein 5. So the kinds of mutations we're seeing in protein 5, protein 6, A and B, and protein 7 are very different influences. So we're excited by this new information, and I'm sure it's daunting for everybody now. We've got new questions to ask and new possibilities. This study presents evidence of genetic variation across 95 known isolates of rose rosette in the form of transitions, transversions, indels, recombination, and reassortment. Our tables show the propensity for nucleotide differences among segments, and that there are different selection pressures acting on the different RNA segments. All RNA segments have examples of transitions and transversions, and most recombination breakpoints were identified in the concatenated sequences, suggesting that there's exchange of segments, but not really breaks within a segment happening. The genetic variation that we saw does lead to amino acid changes, which can affect serology and virus-host interactions. And so we have not been able to develop a clear phylogenetic relationship according to geographic distribution of sequences, because the literature so far doesn't have really specific information about GPS locations of isolates that have been reported so far. So most of this is hearsay, so we can't yet give you that type of information, but I think the information is pretty good regardless. So again, I wanted to thank my collaborators at the beginning of our talk, and we'll be open to any questions. That's all I've got. Yes, Francisco.
Yeah, I'm wondering about the comment you made about RNA5.
Yes.
That is a segment instituted for mutations and gradient systems.
Sure, and I would also invite Ramon and John if you want to answer any of these questions. So the question was about our RNA5. So here on the right in panel A is the amino acid mutations that exist across RNA5 or protein P5. So, okay, perhaps I should rephrase that a little more. We don't know anything about resistance and resistance breaking. This is an example that shows that there's extensive amino acid changes in P5 protein. We don't know the function of P5 protein yet. We don't know the function of most of these proteins yet, but I would want everyone to, if the question that has come back to me has been regarding virus-host interactions, what mutations exist in the genome that could be useful to consider for future hypotheses, I would pay attention to this.
Well, Jeanmarie, one thing we discussed is that a lot of these changes we suspect are due to infection of different host genotypes, because viruses will typically adapt to a different host, and there may be different degrees of selection on each genome segment by differences in the genes between different genotypes. One of the problems there is that many of these 95 sequences are only identified as originating from Rosa hybrid.
Yeah.
And not the actual cultivars. So we can't make that kind of, we can't put a lot of detail on that, but that is our suspicion. And we hope that as more full sequences are deposited, that more sequences will be labeled by the cultivar that came from, so that would help us a lot. But yeah. And if we were to have that information, the breeders could potentially provide us with information about the genetic background of each of those cultivars that would enable us to follow the genes that the host genes that may influence the virus selection.
Excellent point. Thank you very much for adding that. I'll take the question. Yes.
Especially prevalence, high prevalence where it has been very that's actually taking that there are variations in that high rate of high. Also, you get
Yeah. Yeah, I think that's why I piped up earlier about, no, don't want to lose that map. So, but again, the folks, and I would encourage our virologists who are in the room and outside of the room, as you guys are sequencing any component of and depositing that information, please deposit the information about what cultivar or species it came from, because it's quite possible that the divergence is because of the host and not because of geography, and like John said. So
You probably, I guess you don't have an answer. I wonder if anything might be fine.
Ramon, are you here?
I'm here. I didn't hear the question.
So the question was, do any of these isolates come from species roses?
Yes, species cross. So the RF, the one from DC is Rosa fortuniana, where the SW is stormy weather from Delaware. So that was part of the research plot from Tom Evans. So yeah, the one that we have is Rosa fortuniana. When you go to GenBank, as John already indicated, most all of them just say Rosa hybrid. So we're assuming it's knockouts of some sort. I think one of the ones in California has a species name, but it's I don't recall what it was. It's nothing.
Cultivar name, not a species name, isn't it?
Probably, yes.
Yeah.
Right.
We spent a lot of time asking our colleagues if they could disclose their cultivars or isolates or geopositions, and we didn't get that information either. So this is what we've got. Yes.
Great. You mentioned that a lot of these changes can be due to the germ class that the isolate may have come from, the genetics of the rodent.
Yes, that's one idea.
How about the vector?
Yes. We know nothing about the vector.
But you would think since it is native, but it has quite a bit of variation. So, I mean, I'm just wondering if, there are some of these mutations are making it more easier for the vector to transmit it, or, when y'all are thinking host, and I'm sitting here thinking
Vector.
vector.
So I'm going to again defer to my colleagues, Ramon and John, who've been working more on the mite studies. So the question is, how many of these mutations could be actually happening because of the vector?
Certainly could be some, but one hang up there is that we don't yet know the mechanism by which the vector transmits it. Some viruses are taken up and only mechanically transmitted by the stylets. Some go directly to the salivary glands and then back again on the next host. Some have to passage through the gut. At this point, and then back to the salivary glands. And at this point, we have no idea of where the virus accumulates in the vector and what its actual means of transmission is. We are working towards that, but we're certainly not there yet. But there are other factors that may influence that. I mean, there may be differences in mite proteins that interact with the virus that would preferentially increase efficiency of transmission of one strain over another. And also the possibility that different populations of the mite may have different endosymbionts that either positively or negatively affect transmission. And that could be isolate specific, depending on what the interactions are between the virus and mite components.
The other additional, go ahead, John, sorry.
So there's lots of things that we would love to be able to answer and are not in a position to.
To answer directly Mark's question, RNA2 is the glycoprotein precursor, which is the protein that's on the outside of the virion, which the mite would probably see, most likely, as well as the plant. So that might have some effect on either uptake or transmission. So the one on the right here, B. So there are changes there, but not a lot across the whole spectrum of isolates.
And then there are a couple, yes.
So you mentioned in the RDRP, what?
So there's the RDRP in panel A and the glycoprotein in panel B. Okay.
I wonder why is because instead protein by identification, not they are colored their participation in the RDRP compared to the other.
So the question is just to share with everybody, the question is why is the mutation rate in the RDRP so high, given its importance in virus replication? And I would put it another way. It is the longest RNA segment of all the RNA segments. And so polymerases themselves are prone to mutations. And given the length of that RNA segment, it's like 7,000 compared to maybe 4,000 on the, or 1,500 on the RNA5. Because of its length, you are going to have more opportunities for mutations to be introduced. Whether those mutations impact protein function, that's another question entirely.
It's not only the length that can explain that, but also the fact that the replicase has multiple different functional domains, which are essentially separated by spacer domains. And the spacer domains tend to be much more variable than the functional domains. So many of the changes may be in the spacer domains, which probably do not affect function very much.
I want to say that you're correct, John. In our initial looking, just this didn't go in the paper and we didn't analyze it that closely, is yes, many of those are in fact are not in the actual RDRP, helicase, et cetera, domains. So yes.
The sequence then then goes the HDS, but the HDS is not used for the study. You have to say that for time, you have to have 95 grams, and then take an L. Then you do more attention on the variation on the HDS. I'm not wrong. How people submitted there is only HDS. And then put down to 1 meter of it, and you don't get a nice thing.
So the question is, were the sequences that we worked with off of GenBank due to HDS sequencing or Sanger sequencing, the idea that the depth of information would be different with the two different technologies, and so that could influence our data?
That is true. So as Jeanmarie indicated, those sequences that we weren't comfortable with because of their length, those were excluded. And most of those other 95, other than R2, were done by multiplex PCR and then high throughput sequencing of those PCR products, where ours was a high throughput sequencing of total RNA from the infected plant. So in this study, confident that there is depth of reads for each site. You might argue that the multiplex PCR may have introduced errors in that production of the products to be sequenced, but the sequencing themselves were, hundreds of thousands of read depths. So I hope that answers the question.
And those sequences were also published in other reports, so those reviewers let those go through as well. So it was found to be acceptable. Okay. Yeah. Yes. So let me ask my colleagues here who are working on the mite component. The question John and Ramon, are there multiple biotypes of the mite?
I think if you would ask Yanis, the answer would definitely be yes. We don't have the information. Ron could tell you that morphologically there is some variability, but we have not looked at that. I mean, just within a population, we certainly have not been able to compare multiple populations for morphological variability. And so far, we have not done any genetic variability studies, but Yanis does have some information on that score.
I guess that tomorrow when I do the presentation, we will address this issue again. And maybe we can use another samples with similar problems that we have, for example, with the Aerophile mites that attack wheat and the Aerophile mites that attack tree of heaven. We will address this again tomorrow. Keep in mind, and we'll go over again.
Very good.
That's great.
Okay, thank you.